Welcome to the lecture series in food biotechnology. In this series of lectures, we have been seeing how the development in the field of genetic engineering, recombinant DNA technology, and the series of developments which we have seen in the bioprocess can be applied to the field of food production, its preservation, as well as product formation. In this series of lectures, in today's lecture, we are going to see how animal cell culture is usually done and what are its applications in the field of food industry. When we talk about animal cell culture, it's like from a tissue, you are going to isolate a single cell, then you trypsinize that particular cell, and then you use those cells like enzymatic degradation or even mechanical chopping from the tissue, and then formulate a suitable medium. We call this as the primary cell culture. We wait till it forms a monolayer in the culture flask, and then this develops into what we call as the primary cell line, and from the primary cell lines we can actually have the secondary cell lines, the tertiary cell lines, etc. And this cell division will continue up to a finite level depending upon what is the type of cell which we are culturing. The indefinite cell multiplication happens in the case of cancer cells because there they keep on indefinitely dividing. But then, using the applications, what changes can we make in the cell culture and how can we utilize it for the production of certain components which can be utilized in the food industry is what we are going to see today. So, cell culture is a process by which the human cell, the animal cell, or even insect cells are grown in a favorable artificial environment. So, these cells can be derived from multicellular eukaryotic organisms. They can be taken from the already established available cell lines or from the established cell strains. And in the mid-1900s, animal cell culture became a common laboratory technique, but the concept of maintaining a live cell line which is separated from the original mother line or the tissue source, this was discovered in the 19th century. So the idea was there, but what exactly has to be done to maintain this cell culture? What were the problems involved in the maintenance of the primary cell culture as well as the maintenance of the secondary or the tertiary cell lines? These were a big question mark to the scientists at those times. The development of the basic culture media has enabled the scientists to work with a wide variety of cells under controlled conditions. This has played an important role in advancing our understanding of cell growth and differentiation, identification of the growth factors, and understanding the mechanism underlying the normal function of various cell types. See, there are different types of tissues which are present in a multicellular organism, and each tissue actually has a specific function of its own. So the cells of that tissue would be genetically programmed to do that particular function in a more manner, as well as coordinated with the rest of the tissues so that that multicellular organism will function normally. So how do we find out that what are the genes which get activated to perform a specific function? What are the genes which are common to all cell lines? So all those basic details have to be known before we say that this particular cell line I'm going to use to produce this. So those were the questions which were raised by scientists in those times. Animal cell culture has found use in diverse areas from basic to advanced research. And this has provided a model system for various research efforts. The study of basic cell biology, cell cycle mechanisms, specialized cell function, cell to cell and cell to matrix interactions. See, when I say cell is a unit of life, the most single unit of life, which is very important, prokaryotes or bacteria exist as a single cell. So there is no question of it interacting with another cell unless it is for pathogenesis or some symbiotic association. But in the case of a eukaryote, where you have multi-tissue organization, as I said earlier, this individual cell has to communicate with the cells within that particular tissue as well as the cells in another tissue for a coordinated effort. So for that we talk about, so you have the different signaling pathways, signals from the extracellular environment, signals from the intracellular environment, and then how the cell processes it. So all these things together actually coordinately work for the normal functioning of the multicellular organism. So for this, if I have a brain cell or a kidney cell or a lung cell, individually I can culture them and try to find out what is the commonalities between these different cells which grow up, and then in what aspects do their functions differ or the metabolism differ. So for such basic studies, we can use these cell culture. Toxicity testing to study the effects of new drugs. In clinical trials, especially when a new medicine or a drug has been formulated, its effect on, depending upon whether we are going to, the consumer is human being or whether it is an animal, the drug has to be tested before it goes out to the market. So before giving it to humans for consumption, normally it is tested in the cell lines to check the absorption. Say if I say 500 milligram of the cell of the drug I add, how much is actually absorbed by the cell. From that, what is that we call as the bioavailability. Then this 500 milligram, when it is absorbed, see it will be absorbing maybe 100 milligram or 150 milligram from that. So how does it decompose? How does the cell metabolize that? What is its reaction? Is it allergen? Is it toxic? Or does it produce any other adverse reactions? So all those testings can be preliminarily done using cell lines. Then production of viruses for use in vaccine. So you can produce the vaccine, instead of live viruses production, you can use the animal cells. Example, chickenpox virus, polio, rabies, hepatitis B, measles. For all this, live viruses can be cultured in cell lines and we can use them to produce the attenuated vaccines. Gene therapy for replacing non-functional genes with functional gene carrying traits. Okay, this is a very interesting and a new development where when you know that this particular cell lacks a particular gene or that particular gene is mutated causing a deficiency, we can use cell lines to test whether we can transform the cell line. We can introduce the normal or the wild type gene into this cell so that its function is regained. That's called as gene therapy. Then the characterization of cancer cells, the role of various chemicals, viruses and radiation in cancer cells. So for each cancer cell has its own peculiarity. Other than the commonality that cancer cells keep on dividing, each type of cancer has its own metabolism, its own type of activity. So how does it grow? What are the repercussions in the adjoining tissue? What sort of a damage does it cause to the environment? And then if you have a drug against that particular cancer, how does that drug work against this particular cancer cell line? So all those you can study using cancer cell lines from different, which have originated from different sources. The final is the production of vaccines, monoclonal antibodies and pharmaceutical drugs. So cell lines can be used for even that, animal cell cultures. The first mammalian cell culture date back to the early 20th century. The cultures were originally created to study the development of cell cultures and normal physiological events such as nerve development. See, when the idea or the establishment of cell culture was first started, the first criteria or the first priority was given to finding out how the metabolism in an individual cell, what is happening inside. So if I give a stimulus, what happens inside the cell? Which are the proteins which are produced? How does the metabolism process this? So normal physiological events, and even when we are talking about embryonic development, the different developmental pathways. So how does it differentiate? So that was the primary interest when cell culture techniques were developed first. Rose Harrison in 1907 showed the first nerve fiber growth in vitro. However, it was in 1950s that animal cell culture was performed for the first time in an industrial scale. The occurrence of the major epidemics of polio in 1940s and 1950s and the accompanying requirement for huge amount of viral vaccines. So this actually paved the way for the development of cell cultures on a large scale, primarily to produce vaccine against polio. So a huge breakthrough was made during this particular period. The polio vaccine from the deactivated virus became one of the first commercial products developed from cultured animal cells. If you look at the history of animal cell culture development, in 1878, you had Bernard. He established that the physiological state of the cell, similar to the live cell, can be maintained even after the death of an organism. That is, we can maintain even if the organism dies, if you have taken out a cell and you are maintaining it in the culture, it will maintain its characteristic. In 1907, Harrison showed that frog embryo nerve fiber growth in vitro, and he also showed cell entrapment. 1912 had Alexis Carrel. He initiated the tissue culture procedures for chick embryo heart cells, and he used embryo extracts as the cultural media. So when we say animal cell culture, you require specialized media for that. So here he used the embryo extracts for the culture medium, and he tried to grow the heart cells. And for a period of 34 years, he could maintain this. He could passage the cells and maintain them. In 1913, Steinert, Israeli and Lambert grew vaccinia virus in fragments of guinea pig corneal tissue. 1916 had the development of trypsin to suspend the attached cells in culture. 1927, you had the first viral vaccine against chickenpox, the development of the first viral vaccine against chickenpox. And in 1948, single cell culture was established using microscale glass capillaries. This was a huge breakthrough. In 1952, human cervical carcinoma cells from a patient called as Henrietta Lane, these are the ones which we now call as the HeLa cells. The continuous cell line from the HeLa cell lines were established in 1952. And Eagle in 1955, I'm sure most of you would have heard about Eagle's and Dalbeco's medium. The media composition, the established nutrient requirements of cells for the culture, and the defined culture media for cell culture was established by Eagle in 1955. And in 1956, you had Littlefield, who used the HAT medium, that is the hypoxanthine aminoterine thymidine medium, to induce or to select certain specific cells having certain characteristics. In 1961, you had Hayflick and Moorhead. They studied the human fibroblast cell lines and showed that these cell lines were not growing infinitely or indefinitely, but they had finite growth, that beyond a particular number of divisions, they did not exist even in culture. And in 65, we had the first serum-free hams media development. In 1965, monoclonal antibody development happened, and we had the development of the first hybridoma secreting the monoclonal antibody. This was again a major milestone in animal cell culture. And 1977, Genentech, the company, established the first recombinant human protein, stomatostatin, was obtained, purified and obtained and commercialized from cell lines. This was by the company Genentech. And in 1979, Terry et al. Devised gas chromatography system using silicon-etched microchannels. 85 had the production of recombinant tissue plasminogen activators, TPA, in mammalian cells. And also we had the development or the synthesis or the secretion of the human growth hormone from the recombinant bacteria. This recombinant growth hormone produced by the bacteria was accepted for a therapeutic usage in 1985. And this was again a major breakthrough or a milestone in the history of genetic engineering and recombinant technology. In 1986, the first monoclonal antibody was approved by FDA for human use. And again in 86, interferon alpha 2A, Genentech, the company, commercialized the first recombinant protein, interferon alpha 2A. In 1989, we had another company, Amgen, which produced erythropoietin. This was produced in CHO cells, and this was made available commercially. 1992 had again for separation of molecules. See, when we produce recombinant protein, we need to separate them out from whether it is a mammalian cell culture or a CHO cell culture or from E. coli using, whichever is the platform where you are producing the recombinant protein, from among the total soluble protein which is present, your protein has to be separated out, purified. So for that, we had Mans et al. Having a micromachined glass strip electrophoresis for separating the molecules. 1992 had the FDA giving approval for the blood clotting, recombinant blood clotting factor for treatment of hemophilia. This was again, this FDA approval of the recombinant human blood clotting factor again was a major milestone in this case. In 1996, we had another major or very important development in the history of genetic engineering, the production of the transgenic sheep dolly through nuclear transfer technique. So this was actually the first transgenic animal created. So far we have been talking about recombinant protein production, or putting a gene, human gene, into a cell and asking that cell to produce the protein, secreted protein, and purify it. But creating a whole animal was a totally different scenario, and this was succeeded by Wilmot et al. In 1996. 1997 had et al. Activating the microchip device for enzyme assay. 1998, we had the system, again another system development of microfluidic for using PDMS, that is polydimethyl siloxane. In 2000, we had another technological development as a bioanalyzer in the lab. And in 2002, you had Clonaid, the company. They claimed that they have cloned a human baby, whom they called as Eve. Now, the ethics of cloning, human cloning, is a totally different picture. But here, this was again, scientifically speaking, a major achievement. In 2011, scientists discovered that some stem cells treated with certain chemicals could turn into precursors of liver cells. That you can. Stem cells, as you know, are cells which are capable of developing into any type of cells. We talk about the pluripotent stem cells or the embryonic stem cells. The embryonic stem cells are capable of developing into cells of any type. They can demarcate or differentiate into any type of tissue. Whereas pluripotent stem cells, the other kinds, they are already differentiated to one level. So from that level, whatever can be derived will be derived from them. But what are the signals which have to be given for pushing or inducing development was unknown. And this breakthrough came in 2011 to induce the stem cells to turn into liver cells. In 2012, we had more major development in the field of proteomics, and 2012 had the major first gene therapy. As I said, gene therapy is where when you find out a defective gene, if you can introduce the new wild type gene, which is normal, then the patient having the mutated gene will escape from the manifestation of the disease. So this would be extremely personalized because we cannot know which person carries what mutated gene. So if you know a hereditary disease and you know that these people definitely carries this gene, especially if it is a single gene character, we can try to go for gene therapy. So the first successful gene therapy against aging association decline in mice. This was not done in humans, this was done in animals. See, anything related, if it has to be scaled up to humans, first normally has to be tested in cell lines, then in live animals, we normally take mice, then to hamsters, then to monkeys. So like that, the step of development comes. So here, this first, the gene therapy was tested in mice. how do we establish a cell culture? That is the first question. We saw the development so far, but what are the steps to be taken if you need to establish a primary cell culture? There are three major methods which are used to initiate a culture from animals. The first is the organ culture. We take the whole organs from embryos or partially adult organs, and these are isolated whole organs or the partial adult organs are used to initiate in vitro organ culture. So these cells in the organ culture maintain their differentiated character. So this is already differentiated to a particular organ. So the cells will maintain what is the quality of that particular organ. It will not go back. Like in the case of plant cells, we saw that they completely de-differentiate into meristematic cells. Such a thing will not happen here. But what is the functionality or peculiarity of the cells of that particular organ has, that would be maintained in the cells which are growing in the culture. So their functional activity and also retain their in vivo architecture. And they do not grow rapidly, and cell proliferation is limited to the periphery of the explant. So these cultures cannot be propagated for long periods. Organ culture is useful for studying the functional properties of the cells, that is production of hormones, and for examining the effects of external agents, that is if you give drugs or if you give some heat treatment or some other macromolecules, how is this taken up by the cell and what is the reaction the cell has to the drug or to the particular chemical component which you have introduced. So for that, the organ culture can be used. Primary explant culture. Fragments excised from the animal tissue may be maintained in a number of different ways. The tissue adheres to the surface aided by an extracellular matrix constituent, and this can be a collagen or a plasma clot, and it can even happen spontaneously. So this gives rise to the cells migrating from the periphery of the explant. So when you have an explant and it is normally held together by the extracellular matrix, sometimes even the peripheral cells can separate out from this explant. This culture is known as the primary explant, and the migrating cells, we call them as the outgrowth. This has been used to analyze the growth characteristics of cancer cells in comparison to their normal counterparts, especially with reference to altered growth patterns and cell morphology. See, when such cells escape out, especially in the case of cancer cells, they can migrate to other places, right? From the primary or from the node of cancer, the primary node, it can migrate to what we call as the metastasis. So what changes happen there compared to the original cell, or what, suppose a normal cell becomes cancerous, what are the changes which happens there? So for altered growth pattern, morphology, all that can be studied using this type of cultures. Cell culture. This is the most commonly used method of tissue culture, and this is generated by growing the cells, collecting the cells growing out of the explant or from the dispersed cell suspensions, that is, they floating free in the culture medium. So cells are obtained either by enzymatic treatment or by mechanical means, and they are cultured as adherent monolayers on a solid substrate. There are three types of cell culture. One is the precursor cell culture. Here, undifferentiated cells are committed to differentiate. So, undifferentiated cells which are not being destined that I will become this particular tissue or I will become this particular tissue. When you culture them, they will differentiate to become that particular tissue level. Then differentiated cell culture. Here, completely differentiated cells that have lost the capacity to differentiate. So, these will be maintained as such, but they will not differentiate further. Then stem cell culture, where undifferentiated cells that undergo or they go on to develop into any type of cell. So stem cells, as you know, can develop or differentiate into any type of cells, but in culture we are maintaining them only as stem cells. We are not asking them to differentiate. Cells with a defined cell type and characteristics are selected from a culture by cloning or by other methods, and then this cell line becomes a cell strain. Monolayer culture. The monolayer culture is an anchorage-dependent culture of usually one cell in thickness with a continuous layer of cells at the bottom of the culture vessel. The second type is a suspension culture. Some of the cells are non-adhesive, meaning they don't stick to each other, and they can be kept in mechanically mechanically kept in suspension, unlike most of the cells that grow as monolayers. See, usually when we take animal cell cultures, they grow as monolayers. But some of the cells will escape out from this. They don't stick on. So then they can be maintained as a suspension culture. Example, leukemia cells. This offers numerous advantages in the propagation of cells. Passaging. Passaging is the process of subculturing cells in order to produce a large number of cells from pre-existing cells. So you have separated out a cell, we have established a primary cell culture, and when we make multiple passages, that is keep on changing the medium and maintain it in an aseptic condition to produce so that the cells will divide. So from the initial low number of cells, we can get 10 to the power of n number of cells. So this production, this by passaging, it is a subculture where from a lesser number of cells, we are going to multiply and get more number of cells. This subculturing produces a more homogeneous cell line and avoids the senescence associated with prolonged high cell density. See, when you don't subculture, after a particular point of time, the media would become exhausted and there would be too many number of cells present in the particular media, which can induce aging. So when we keep on subculturing, that senescence associated changes can be avoided, and we will be getting more number of or a high density of cells, more number of cells. Splitting cells involve transferring a small number of cells into each new vessel. So every subculture, we will aliquot some from the original culture and put them into fresh so that they will again have more media and more nutrients to keep on dividing. After subculturing, the cells may be propagated, characterized and stored. Adherent cell cultures need to be detached from the surface of the tissue culture flasks or dishes using proteins. Proteins secreted by the cells form a tight bridge between the cell and the surface. See, when you have an adherent cell culture, the cells actually attach themselves to the substratum, and this attachment is taken care of by the proteins secreted by the cells. So between the cell and the surface, they have a tight binding or a tight bond. So to break the protein at specific places, we use what we call as a trypsin EDTA mixture. Trypsin is proteolytic, so it hydrolyzes the pepsin digested peptides by hydrolysis of the peptide bonds. EDTA sequesters certain metal ions that can inhibit trypsin activity and thus enhances the effect of trypsin. So if you see what are the basic applications of animal cell culture, this can be used, animal cell culture can be used as a model system because instead of using animals to test, we can use the cell lines as a model to know the effect of the particular drug or the treatment. Then if it succeeds and if there are no bad effects or toxic effects, then we can take it to the animal models. Second is test the effect of drug on cell growth, which is the toxicity test. Test the therapeutic effect of drug on certain cancer cell lines. Then they can be used for tissue engineering, can be used for the production of vaccines and monoclonal antibodies. Then study for the replication and the life cycle of the virus, especially if you have a virus attacking a particular cell. How does the virus take over the cellular mechanism? What does it do to replicate inside the cell? How does it affect the cell cycle? So for all that, we can use the cell culture model, embryo culture, production of therapeutic proteins. So these are the widespread applications of animal cell culture. Quantitation. So once you have established a cell culture, we need to characterize the cell growth and to establish what we call as the reproducible culture conditions. So this can be done by using hemocytometer, electronic counters, or in some cases by DNA or protein concentration. Many culture methods that start with a dispersed population of cells encourage the arrangement of these cells into an organ-like structure. So these type of cultures can be divided into two basic types. The histotypic culture is one, where we see the cell-to-cell interactions. See what type of interactions are seen between two tissues would be seen in cell-to-cell interactions in the case of a histotypic culture. And for this, we use an appropriate extracellular matrix content as well as the soluble factors. And we also grow the cell cultures to very high cell densities. So how do we establish this? Growing cells in a relatively large reservoir with adequate medium to growing cells at a very high concentration on agar or agarose or estered aggregates or what we call as the spheroids, and or 3, growing cells on the outer surface of hollow fibers. The cells get seeded there on the outer surface and the medium is pumped through the fibers from the reservoir. Histotypic culture can be established by different methods where we grow cells in a relatively large reservoir with adequate medium, or growing cells at a very high concentration on agar, or growing cells on the surface of hollow fibers. How we do this and what are the other types of cultures which can be done would be dealt with in the subsequent lectures. Thank you.